Disease prevention using CRISPR. CRISPR-Cas9 is a powerful genetic tool that allows scientists to edit DNA. It is based on a bacterial immune system that enables bacteria to defend themselves against viral infections. This system has been adapted for use in genetic engineering and has been hailed as a breakthrough technology that has the potential to revolutionize the field. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which are short, repeated sequences of DNA that are found in bacterial genomes. Cas9 is a protein that is part of the bacterial immune system and acts as a molecular scissor that can cut DNA. This allows scientists to edit genetic sequences with precision and accuracy, enabling them to make changes to existing genes, inactivate them, or even create new ones. It works by using a set of molecules known as guide RNA to find and target a piece of DNA within a cell. The guide RNA is made up of both a tracer RNA, which helps the Cas9 protein find and bind to the target DNA, and a CRISPR RNA. This sequence is complementary to the target DNA sequence, which allows it to bind to the DNA at the precise location where the researchers want to make a cut. Firstly, Cas9 searches for the complementary DNA sequence. Then the guide RNA hybridizes with the DNA. The nuclease domains on the Cas9 protein then cleaves the DNA that results in a double-stranded DNA break. Once Cas9 creates a break in the sequence, DNA naturally repairs itself, but this leads to mutations and leaves the DNA non-functional. Additionally, breaks allow scientists to insert, delete, or modify the existing genetic material. This process is called editing, and it is highly precise and efficient, allowing scientists to make changes to specific genes or even create entire new genomes. CRISPR is used to edit DNA mutations associated with genetic diseases. It can help cure/treat a number of genetic diseases, ranging from blood diseases to cancer. The ability to change DNA sequences in cells virtually and at any position enables its therapeutic application. Examples of some of the first diseases scientists are tackling using CRISPR include, but are not limited to, cancer, blood disorders, cystic fibrosis, and muscular dystrophy. Although CRISPR can't necessarily cure cancer, it is a valuable tool in cancer research as it can be used for things such as identifying genes, screening for cancer drivers, and identifying proteins that act as targets by cancer drugs and diagnostics. For example, China began a new study testing the use of CRISPR to modify immune T cells extracted from individuals with lung cancer. CRISPR has also been used to improve the safety and efficacy of cancer immunotherapy. In terms of CRISPR and blood disorders, sickle cell disease is the most common blood disorder targeted by CRISPR treatment. Essentially, the excess cell therapy is currently being tested, and it consists of getting bone marrow stem cells from patients and using CRISPR to make fetal hemoglobin, which is much better than the conventional form of hemoglobin for adults. These modified cells would then be reinfused into the patients. CRISPR can individually edit the mutations that lead to cystic fibrosis. There are also therapies of treatment potential using CRISPR, but they still remain in the research phase. Research in mice has shown that CRISPR can be used to fix the genetic mutations behind the disease of muscular dystrophy. In terms of CRISPR and muscular dystrophy is a genetic disorder caused by the mutations in the gene encoding for the protein responsible for contraction of muscles. The disorder is typically present in early childhood and begins with proximal muscle weakness. Due to the complex underlying genetics of the disorder, there is no known cure for muscular dystrophy. Treatment aims to control symptoms in order to improve an individual's overall quality of life. CRISPR has recently been explored as a treatment mechanism for muscular dystrophy. The CRISPR system consists of two components: a single guide RNA, guiding Cas9 to a specific site in the genome containing the complementary sequence, and a CRISPR-associated endonuclease. This acts to eliminate mutations associated with muscular dystrophy and restore the protein expression throughout cardiac and skeletal muscle. That being said, several aspects of muscular dystrophy make CRISPR a possible therapeutic approach. The method of taking muscle stem cells from diseased patients and using CRISPR to correct those faulty genes, and then injecting them back into the muscles to allow for proliferation of new muscle tissue has been pursued. Although researchers were only able to show this method works in mice with muscular dystrophy, it is a suitable idea for patients, but it is unsuitable for the actual treatment of patients due to the unknown safety and efficacy. In terms of the limitation of CRISPR requires an appropriate vector when delivering the nuclease encoding genomes and guide RNA in vivo during the process of gene editing. The employment of a very small Cas9 gene could significantly negatively affect the efficacy of the process. It is to be noted that in vivo delivery of CRISPR-Cas components can trigger immune responses against the Cas protein, and that there is even some evidence of potential risk of development of cancer in patients. The utilization of any other forms of delivery requires further research, analysis, and optimization. Additionally, ethical dilemmas may arise regarding treating genetic disorders in patients. Numerous countries, such as China, have forbidden the use of germline gene editing. Furthermore, the potential for genome editing in embryos instigates fear and mistrust in science and researchers by the public and general non expert audiences. CRISPR, being a powerful gene editing technology, allows for precise and efficient editing of genetic material. The ongoing research and development could lead to groundbreaking discoveries and applications. The future steps of CRISPR lie in human health, agricultural, and livestock production. In terms of human health, CRISPR can be used to make precise edits to the genetic code of cells, allowing researchers to correct or modify the specific mutations responsible for genetic diseases. For example, in a clinical trial for sickle cell disease, researchers are using CRISPR to edit the genes responsible for the disease in patients' blood stem cells with the goal of producing healthy red blood cells. Furthermore, CRISPR can also be used to insert new genes into cells, which could be used to treat genetic disorders caused by a missing or non-functional gene. For example, researchers are exploring the use of CRISPR to insert a healthy copy of the CFTR gene in patients with cystic fibrosis. In the field of agriculture, CRISPR can be used to make precise genetic modifications to crops, such as increasing their resistance to pests and diseases or improving their nutritional content. For example, researchers are using CRISPR to develop crops that are more resistant to drought and other environmental stressors, which could help to improve crop yields and food security. And lastly, for livestock breeding, CRISPR can be used to conduct genetic modifications to livestock, such as increasing their resistance to disease or elevating their meat quality. For instance, researchers are using CRISPR to develop pigs that are resistant to African swine fever, a devastating disease that has caused significant losses in the global pig population. In summary, CRISPR is a promising tool for gene therapy, agricultural application, and livestock breeding, with the potential to correct or modify the underlying genetic mutations. While there are still some challenges to overcome, such as delivering the edited genes to the appropriate cells in the body, regulatory and consumer acceptance, persistence in research and development will allow CRISPR to reach its full potential. We hope this video helped you understand disease prevention using CRISPR. Make sure you pause the video on the next 2 slides and answer the questions to test your learning. Thank you for watching. What is CRISPR short form for? Clustered, regularly interspaced, short palindromic repeats. What are some examples of diseases CRISPR can help treat? Cancer, blood disorders, cystic fibrosis, and muscular dystrophy.